So today we will continue the inhibition process. The last class we discussed about competitive inhibition. We just discussed about the mechanism, number one. Number two, the general flow chart of the mechanism. Number three, the graph, that is Lineweaver-Burk plot in case of competitive inhibition. So today again we start with, so this is just a recap. When it comes as a short note or question, whatever, long note, whatever it is, competitive inhibition or types of inhibition. Okay, so you have two options. You write the inhibition, types of inhibition are of two types: reversible and irreversible. The reversible one are of three types: competitive, uncompetitive and non-competitive. Competitive, uncompetitive and non-competitive. Irreversible, separate. Alright. So under the competitive inhibition, you must first explain what do you mean by competitive inhibition. In one sentence only. In competitive inhibition means the substrate enzyme interface is similar to that of the inhibitor, right? And it competes with the substrate for the same active site, right? Next, you must write this flow chart: E plus S, both side arrow, ES plus E plus P, E plus I stands for inhibitor, forms EI, and definitely EI does not give rise to any product. So either you can just write one arrow P and then cross the arrow, right? Or you can just leave it like this. Ki bola বুঝতে পারলে? Fine. Next আমরা দেব হচ্ছে এইটা কেন দেব না? Why we are not giving this graph? Which one is easier to calculate from? From this graph or this graph? Linear one. So if the graph is a straight line, it is always easier to find out the values. Whereas if it is hyperbolic means, again we have to go for density differential calculation. Okay. So this double reciprocal plot or Lineweaver-Burk plot is the plot of choice for physical calculation of any enzymatic reaction. Correct? Here we have only two axes X and Y. Correct? So this red line is without inhibitor. Why? Correct? See, nice color coded, huh? And the blue line stands for with inhibitor. This is I1, I2, I3. Means what? I3 concentration is greater than I2. I2 concentration is greater than I1. So you are increasing the concentration of inhibitor. You need not to draw all the three lines. You can একটা line draw করলেই হবে. You can draw only one line to explain the mechanism. Okay? But remember these points, the line should coincide with y-axis at the same point where the red line coincide. This concurrentness depicts what? That there is no change in the Vmax. That means what? The enzyme's capability to convert substrate to product is not altered in case of competitive inhibition. Then what is altered? If you can recall, I told that enzyme substrate interaction is a dynamic interaction. It is not like that one enzyme is sitting there and one substrate and looking at each other and it will happen. No. It is not like that she will carry a rose and I will take it and then reaction will happen. No. It is like she will carry a rose and come to me again and again, and I will go back again and again. One fine day, at some special moment, what is the special moment? When the energy is equivalent to activation energy. Then she will give and I will accept the rose. Correct? So that particular moment when the energy dissipated by the collision of substrate and enzyme will exceed the energy required for the activation state or to achieve transition state, then the reaction will take place. Same thing will happen in case of inhibitor. When the inhibitor is a competitive inhibitor, the inhibitor is structurally similar to substrate does not mean the whole inhibitor is same. They are not twins. They are not at all twins. How different they could be? Malonate is a competitive inhibitor for succinate dehydrogenase. What is the actual substrate? Succinate. What is the difference between malonate and succinate? Malonate is a 2-carboxylic dicarboxylic acid containing 3 carbon atom, and succinate is dicarboxylic with 4 carbon atom. So what is the structural similarity? Nothing. See, this is malonate, not malate. Yeah? Here, different. It becomes right. So anyway, so malonate is 2 bicarboxylic acid with 3 carbon acid, which is structurally similar to that of the succinate at this part only. The whole molecule is not same. Clear? So it is the structural similarity lies at the enzyme inhibitor or enzyme substrate interface where they actually shake hand. That's only hand part. The rest of the body is different. Fine, this graph is clear. Now here see the KM. This is normal KM and this point is approaching towards origin. Does it mean it is decreasing or increasing? Increasing. Why? Because it is inverse. Correct? Moreover, negative. One more example is this. One example I gave here. Another example is this. PABA, para-amino benzoic acid. It is required for synthesis of folic acid. PABA is required for synthesis of folic acid. Now the sulfur drug, that is nothing but this one, sulfonamide, has a structural similarity to that of the PABA, correct? So instead of PABA, if this compound, which is used as antibacterial agent, participate in the formation or biosynthesis of folic acid, then folic acid will not be synthesized. Remember, folic acid is one of the vitamin, water soluble vitamin, and our body cannot synthesize it. Means mammals cannot synthesize it, the eukaryotes cannot synthesize it, but the prokaryotes. Bacteria can synthesize them. So in order to kill bacteria which has harbored in our body, we should stop the inner metabolism of the bacteria. So our antibacterial agent should target the inner machinery of the bacteria itself. So we will design a drug which is similar to that of the PABA and interfere the bacterial synthesis of folic acid. What is the importance of folic acid? It is required for synthesis of DNA. So without DNA, bacteria cannot perpetuate, cannot survive, cannot replicate. Whatever bacteria was there, it will get old and it will die, but new bacteria will not be synthesized. So bacteria will die in presence of or in absence of folic acid or in presence of sulfur drugs. This is called sulfur drugs, which contain sulfanilamide. Right? So sulfanilamide is structure similar to PABA, para-aminobenzoic acid, which is a structural component of folic acid required for bacterial biosynthesis of folic acid, in absence of which bacteria succumb to death due to কি বললাম এটা কি বললাম বল যেটা বললাম পুরো বল হ্যাঁ ফল উইদাউট ফলিক অ্যাসিড হ্যাঁ ব্যাকটেরিয়া উইল নট গ্রো তাই তো বাহ কেন ডিএনএ ড্যামেজ হয়ে যাবে তৈরি হবে না বুঝো নেক্সট কামস মিথানল পয়জনিং দিস কোশ্চেন কেম দিস ইয়ার এই বছর এসছে Methanol poisoning. You have any idea about how alcohol is prepared? Anyone? Dari bolo? Alcohol, liquor, mod. By distillation. Distillation of what? Food you cannot distill. Food has to be processed. What is the name of the process? Yes. Fermentation. The fermented product. The sugar solution is fermented. What do you call that reaction? Does it occur outside or inside your cell? Huh? Bolo. Does it occur outside or inside your cell? Right? Dara. Outside the cell. Then how come it is anaerobic? Outside your cell, so much of oxygen is there. Huh? Outside the cell, compact. The cell is within the cell. Huh? Cell in jail. Cell in jail. Huh? Anyone? Bol. জোরে বলবি না আমি তো কাজে যাব জোরে বল when the cell has no machinery to utilize oxygen, or cell is under stress not to utilize oxygen, then anaerobic respiration takes place. But what the reaction we are talking about is not anaerobic respiration. It is a fermentation process. Why do you need fermentation process? Because our cell cannot metabolize polysaccharides. We can absorb and metabolize only monosaccharides. Only monosaccharide, not even disaccharide. Correct? But when you take the molasses, it is what? Either a mixture of polysaccharide and disaccharide. So we need to break it down to monosaccharide. How do you do that? We take the help of an organism called yeast. Yeast produce and secrete certain enzymes. What do you call it? Call that enzyme. Zymase. The zymase is secreted outside the cell. Where it is thriving, the solution, sugar solution. You take a grape juice, add a little bit of yeast, let it perpetuate, provide required temperature, humidity, etc., etc. And then it will secrete zymase, which will convert sugar solution into ethanol. But not only ethanol. There are many yeast which will also produce propanol, butanol, methanol. When we distill, if we increase the temperature, we get methanol instead of ethanol as a product. Product is a mixture of all oils. When we distill, a temperature, if it is little bit higher than the ethanol required for ethanol separation, then it separates out methanol. When the temperature is below than required for ethanol separation, it forms propanol. Now, if the temperature is less, consumption of propanol is not harmful. Consumption of propanol is not harmful. It will not give you a kick, but it is not harmful. But consumption of methanol is harmful. That is why, where the distilleries are not properly regulated under the observation of government or any other body with strict regulation, it might produce spurious alcohol, and which mostly contain methanol. The problem is, উল্টো উল্টো করছি কেন? কি হলো কি? Right. Now this acid is formic acid, and this is what? Huh? Jore. If I write this, okay, now tell me which one do you consume? Vinegar. So is it harmful? No. So if we consume ethanol in liver, it is converted to, okay, there are many other pathways anyway, it is like that. These reactions, both the reactions produce lot of and the name of the enzyme is alcohol dehydrogenase, aldehyde dehydrogenase, both require zinc as cofactor and both are located in the liver. That is why when we consume alcohol, our liver gets affected. Right? But because this is very costly, so we have purchased alcohol at a cheaper rate with a risk of finding this. Now this one has been prepared in a distillery where temperature is not maintained properly. So the content is more methanol than ethanol. Now this one will produce this. Okay, no problem. So far so good. But when it produces this one, can you tell me what happens when this one is injected in your body? Have you ever felt it, experienced it? Anyone? Cable. Antibite. Who has not taken antibite in your life? Is there anyone who has not taken antibite? Who has never been bitten by ant? Anyone is there? No one? So it is a acid present in the antibite. The red ants. The black ants are good, black guys. Red guys are not good. So this formic acid is such a small amount under the skin, it produces so much of pain. Just imagine if it is produced in amount of ml. This is micro ml. And now it is being produced in our liver in ml. What will be the condition of the liver? Yeah. Tata, bye bye. You have seen the meme? So, gaya, tata, bye bye. So, liver will go on. Now, our body can get rid of formic acid, but it will require some time. Right? So, what happens when a patient comes with this formic acid actually it causes harm to the neurons, nerves, mostly optic nerve. The optic nerve gets affected by formic acid. That is why you might have heard that during this period of time, winter period, in rural areas, the underdeveloped areas, people have succumbed to poisoning with spurious alcohol and they have lost their vision. Have you heard of this news ever? Anyone? কখনো শুনেছো? না আমি বললাম তখন শুনলে। এর আগে শুনেছো কখনো? কোথায় শুনেছো? তোমার তো নিউজ পেপার পড়ো না। There is no word called newspaper in your life. How many of you read newspaper or ever read newspaper? How many of you has read newspaper only for once in your life? Okay, twice. It's fact. It's a fact. For us, reading newspaper was a lesson for learning English in our time. I was from Bengali medium. Yeah. Different newspapers to come for different types of English. Yeah. Nowadays, neither your Bengali is strong, nor your English is strong. Other language is not buttoned into me, so I cannot take the charge. Anyway, so this formic acid is very dangerous for optic nerve. We must slow down the synthesis of formic acid so that our body gets time to get rid of it slowly. So what we do? We give patient alcohol, pure ethanol. Most of the times it is written consumption of alcohol is harmful for health, but being doctors, you will prescribe them alcohol. If patient is unable to take, you have to give them alcohol forcefully. Okay? So that patient can survive. So ethanol is available, pure ethanol is available in emergency because it's an emergency condition. There you have to give them, if the patient can drink, okay, fine. Or you have to give injection for small amount, not sudden huge amount. The patient again will take up the speed. Yeah. So alcohol, slowly, alcohol goes and compete with the same enzyme. So this process is slowed down. Again, this process is slowed down. Right? So now 2 molecules, 3 molecules of this will be produced, but only 1 molecule of this will be produced. So our body will get some time. We give the patient a normal saline. In with the saline, it will drain out to urine, the formic acid. So patient will get some, the body will get some time to get rid of formic acid naturally. Nothing else. This is not medication. This is called therapy. Alcohol is not a medicine. But the question might come, write down the use of alcohol as a medicine. Medicinal value of pure alcohol. So this is that. Non-competitive. Now non and uncompetitive, very much uncomfortable. These two are pretty confusing. And the confusion has increased further with some books who themselves confused it. Books itself are written wrong. How do you know which book is written wrong? You are reading for first time. So here you have to trust who? Not the Almighty, me. For at least this poor portion, you have to trust me, the person standing in front of you. This is for non-competitive inhibition. See the graph, flowchart. Tell me, how do you describe this flowchart? Anyone who is very good in English and who has a very loud voice, robust voice. Either you stand or I will make you stand. Jival. Describe this flowchart. What do you understand by this flowchart? Anyone? Come on, hurry up. Anyone? क्यों ना? अरे तू बोल. Go. এইটা দেখে বল, এইটা দেখে বল, এই যে ফ্লোচারটা বানানো হচ্ছে, এটা মানে কি, এটা বুঝিয়ে দে, মানে বল। বাংলা সব ভালো, মদ ছাড়া, বল। Correct. Enzyme can bind irrespective of the binding with the substrate. ফ্রি এনজাইম বাইন্ড করতে পারে, কেন? তার কারণ হচ্ছে এনজাইম এবং সাবস্ট্রেট, ইনহিবিটর এবং সাবস্ট্রেটের বাইন্ডিং সাইট আলাদা। কথা বলতে পারিস না? পার্টিসিপেট না করলে কি করে হবে? ঘুমাচ্ছিস বসে বসে ক্লাসে? একে বলে মাছের ঘুম। মাছ চোখ করে ঘুমায়, তোরা চোখ করে ঘুমাস। বছরের প্রথম সিজনের শুরু, ঘুমাচ্ছিস। এত বসো বসো বসো। So enzyme inhibitor and substrate can bind with the enzyme at different sites. They do not compete with each other for attachment with the enzyme. That is why inhibitor can bind to free enzyme or inhibitor can bind to enzyme substrate complex. Whatever it happens, it forms ESI, EI plus EI plus S, ESI, ES plus I, ESI. But ES forms E plus P, but not in case of ESI. ESI cannot produce any product. Clear? Follow? So if we see this reaction flowchart, then our graph will be like this. This graph shows what? In competitive inhibition, graph showed that Vmax remains unchanged, Km increases, right? Here, Km remains unchanged, Vmax if it goes higher the x y axis, it is increase or decrease? One by 50 and 1 by 25, which one is smaller? Okay? So Vmax decreases, Km remains same. Why Km remains same? Because they do not compete for binding to the active site. Sites are different, two chairs are different. Why Vmax? Because it is inhibitor by nature of its substance, inhibitor, it must inhibit the reaction. So Vmax will decrease. If Vmax doesn't decrease, it will not be called as inhibitor, it will be called by activator. Why Vmax is decreasing? Because it is inhibitor. If it had increased Vmax after binding, then it would be called activator. Simple. Done? Graph আঁকা হয়েছে? খুব সিম্পল, একটা ফ্লো চার্ট, নর্মাল এক্সপ্লেনেশন, ফ্লো চার্ট। তুই যে লিখছিস না কেন রে ছেলে? দেখা গেছে এটা? বাবা, অত ছোট ছোট করেছিস কেন? হ্যাঁ, এক্সাম্পল পরে আসছি। তাই না? এবার বেশ আনকমফোর্টেবল জায়গায় চলে এসেছি, আনকম্পিটিটিভ ইনহিবিশন। তাই না? Things are getting complicated now. See, substrate binding site, inhibitor binding site. What is the difference? See the last flowchart of non-competitive? Huh? Just cover the left hand side horizontal reaction line. देखो, E+I EI +S ESI, just cover the left hand side. It becomes uncompetitive. That is what? Inhibitor can bind only when the substrate is already bound. In non-competitive, irrespective of binding of substrate, it will bind. This is the flowchart. Followed by flowchart, there is Lineweaver-Burk plot. How it would be, the graph? Any idea? Anyone who is good in physics or mathematics? What is important? See, parallel. The lines are parallel. As you increase a concentration of inhibitor, lines become parallel to that of the graph of without inhibitor. So in both the cases কি হচ্ছে? Because it is inhibitor, Vmax always decreases. Vmax cannot increase because this is inhibition. So Vmax always decreases. And what about Km? Km? Increase or decrease? Suppose this is 10, this is 20. So 1 by minus 1 by 20, and this is minus 1 by 10. Increase or decrease? K? Which one is greater? Huh? এটা এটার মানে হচ্ছে কত? মাইনাস 0.1 বড় না মাইনাস 0.2 বড়? হ্যাঁ, এটা কি আসবে? 0.2 আসবে তো। আমি বললাম না আমি অঙ্ক তো ভালো না। এবার ঠিক আছে? হ্যাঁ, কোনটা বড়? Both Km and Vmax changes. Simple. এই অঙ্ক খুব কঠিন জিনিস, তাই না? Mathematics is very difficult. Irreversible. Irreversible inhibition means where the inhibitor permanently harbor into the active site. It comes as a guest and then it becomes, then it replaces host. Like the Britishers came to India as a trader, then became ruler. Similarly, these people, inhibitors, they come to enzyme, they bind to the active site, whatever may be the reason. They might be structurally similar, they might be more in concentration, they might be through induced fate, they might be through complementary transition state, whatever it may be the mechanism, they will bind to the active site and they will bind permanently. How? There is only one process by which they can bind permanently, that is covalent bond. So once a covalent bond is formed, they are permanently bound. Sometimes they cannot bind in their native state. So what they do? They first occupy the active site, then they are changed to a new product. This product binds permanently, not the initial one. The changed product binds permanently to the enzyme. This kind of inhibition is called suicide inhibition. Suicide inhibition. One such example is penicillin, which is used to break down the bacterial cell wall. Bacterial cell wall. One more example for this irreversible, write down. বলো, ফুল নাম বলো। হ্যাঁ বলে যা, বলে যা, পড়ে আসিনি, বলে যা। তারপর বলে যা। ডাইআইসোপ্রোপাইল ফসফোফ্লোরিডেট, DIPF কি করে? It inhibits serine proteases. Serine protease means, name some protease, say chymotrypsin, correct? Trypsin. এই ট্রিপসিন, কাইমোট্রিপসিন, এই এনজাইমগুলোর অ্যাক্টিভ সাইটে সেরিন রেসিডিউ থাকে। The proteases or proteolytic enzymes containing serine residue at their active site are called serine proteases, and they are inhibited by DIPF. How? The DIPF goes and binds to serine irreversibly by forming a covalent bond, and the enzyme is inhibited forever. The molecule is wasted, so it cannot digest anymore. Clear? One example. Another example is এটাও এসছে পরীক্ষা এবার এটাও এসছে পরীক্ষা অ্যালোপিরিনল কি করে it inhibits xanthine oxidase, the enzyme required for production of uric acid from AMP, GMP, है ना purine nucleotide. So breakdown of purine nucleotide forms uric acid. The last enzyme in this reaction is catalyzed by xanthine oxidase. Last two steps. Xanthine oxidase is inhibited by allopurinol. Allopurinol sits at the active site of xanthine oxidase and it forms alloxanthine. Now allopurinol does not bind so tightly with the xanthine oxidase active site, but alloxanthine. Alloxanthine binds very tightly and irreversibly at the active site of the xanthine oxidase. So now this process is called key. Right? What else? Folate convert to dihydrofolate, convert to tetrahydrofolate by corresponding reductase enzyme. What is the name of this one? Folate reductase. Name of this enzyme? Dihydrofolate reductase. Both require NADPH. Correct? Now, this particular step is inhibited by MAT. কি হলো How? By competitive inhibition. Okay? That is why these drugs are used as what? Now, this process is required for perpetuation of or synthesis of folic acid is required for what? For thymine. Thymine is required for synthesis of DNA. Huh? Right? So, without DNA, our cell will die. And so as cancer cells. Who requires more DNA to be synthesized? Normal cell or cancer cell? Cancer cell. Because it is more, replicating more at higher rate. So, who will suffer more? Cancer cell. If we stop the supply of folic acid to the growing cell, the cell will die. So, these are used as correct? But the problem is what? If you just give the tablet like this methotrexate to a person, then it will reach all the cells, including normal cells. There are plenty of normal cells whose turnover is very high. For example, hair follicular cells, blood producing cells, RBC, and intestinal luminal cells, Rajbora cells. So, what will happen? Improper use of these will cause damage to those. The person will be anemic, person will lose hair, the person will have indigestion or improper absorption, celiac disease, sprue-like disease, malabsorption syndrome, right? So, person has to be given IV supplementation of water-soluble vitamins, fat-soluble vitamins, so that cells can survive. That's all. With this, we come to the end of enzyme chapter concerned to first paper only. The chapter is very big. The second paper we will discuss, part of the second paper we will discuss later. Any questions so far? Any doubt? Then write down the important questions from the chapter. Classify enzymes. Give examples of specificity of enzyme specificity. Give examples of enzyme specificity. How enzymes act. Role of metal ion. Role of metal ion in enzymatic activity. Describe different types of inhibition. Explain with examples. Explain why the following. সব করে চলে এসেছে এবার পরীক্ষায়। Allopurinol used in treatment of gout. Explain why. Question আছে then explain why today. Allopurinol is used in treatment of gout. Explain. Okay, next question. Methotrexate used as anti-cancer drug. Hello? আরেকটা কি বললাম? Ethanol in methanol poisoning. Use of ethanol in methanol poisoning. What are serine proteases? Explain inhibition by covalent modification. Explain inhibition by covalent modification. Describe different techniques. For a question, describe different techniques to find out, to explore active site of enzyme. To explore active site of enzyme. Okay? आगे क्वेश्चन था covariant modification, inhibition by covariant modification.